Hello, everyone, and welcome to the Laboratory Automation and Testing virtual event. My name is Mei He, Assistant Professor from the University of Kansas, and we'll be presenting on the 3D microfluidic technology for empowering the biomedical research. Please feel free to submit questions during my presentation, and I will follow up with you by the email. So, for the three-dimensional microfluidics, and we all know they have the three-dimensional micro nanostructure implemented into the microfluidic device. So as the world we are all living in is essentially three dimensions. So if we look at all the objects present in this world, they all have the height, width, and the depth. So this three-dimensional geometry actually gives a big impact to the surrounding environment. It's also true in the micro scale. So the 3D micro and the nanostructures have the potential for engineering the chemical or the biological system with the fundamentally new characteristics and the functionalities. So I want to introduce some new discovery that we had for this three-dimensional influence on the biomedical research. I want to start with the first fundamental introduction of the microfluidic technology, so which is a device and a technology that can precisely control and manipulate the fluids, cells, and the molecules geometrically constrained in a small network of channels with dimensions from tens to hundreds of the micron. So micron is really small scale. If we look at the bigger scale, that's a very typical scale, like 10 millimeter, for a cell or the molecule to transport through such 10 milliliter length, they will take more than 500,000 seconds to pass through 10 millimeter, which actually equal to the 6 days. It's super long. But in the micro scale, this transport process can be much faster. Usually in the 10 micron scale, for a cell or the molecule to transport through, they only take 0.5 seconds. So it's super fast. That's the real situation in the biological system. So in our body, the cells and the blood vessels are in the micro scale. So this biological reaction and the event can happen so quickly. So in this microfluidic technology, they handle molecule cells in the micro scale that actually provide a lot of advantages, like the high resolution for the single cell, single molecules, the high sensitivity detection of the molecular cells, and very high efficiency in terms of mass transfer. And the sample consumption is very low because it only handles the microliter range of the fluids, and the entire device is designable, portable, and disposable. So here I want to make a new introduction to the three-dimensional microfluidic chips. For the past few years, we all mostly focused on the 2D planar microfluidic chip as the one very typical standard microfluidic planar chip shown in the middle of the top panel, which is a serpent channel with a zigzag planar channel geometry for the fluid mixing. And if we design this planar microfluidic channel into the three-dimensional structure as we show in the top left, there are also zigzag structure in the microfluidic channel, but there are change along the z dimension. So the change in the depth create more structure and introduce the fluid to change the flow path in the z dimension. And we can notice through this constant simulation that this mixing process can be substantially changed compared to the planar chip. So if it's just a single straight planar channel, and as we see from the bottom left, there are two fluids mixed through this constant simulation. There's no fluid mixing happen at all because in the micro scale, the laminar flow is dominant, and the mixing process completely depends on the diffusion process, which is slow. But we generate the three-dimensional microstructure, which can generate local turbulence and substantially enhance the mixing process. So the mixing can happen right away in very short time, in few seconds, and which is much efficient compared to the planar chip with the classic serpentic channel, as we show in the middle of the bottom of simulation. We may well compare such big influence in terms of the three-dimensional change of the geometry and see the influence on the mixing process. So we designed a planar chip with stack ring, which is a planar stack ring structure. And through the simulation, you can see the two flow goes through the Y-shaped channel, and the two flows actually do not mix at all. But if we simply turn the stack ring to the 90 degree, so which change the third dimension, generates the local change on the third dimension with the Z depth change. And we also prove this by the experimental demonstration. Just have two fluid dyes, one is in the blue color, the other one is in the red color. So if we introduce those two flows into the planar stack ring microfluidic mixer, and there's no mixing happen. And so as we simulate by the Councilor simulation, so the video actually going to play to show this mixing process. So you're going to play the video. So as we simulate exactly, and because there's no change on the third dimension of the depth, the two flow mix, and they still keep the interface and won't be able to generate any turbulence in the microfluidic channel. So the mixing cannot happen in such short time, and it totally depends on the diffusion process. So as we demonstrate through this video, the geometry and the scale dimension and the size of the channel are exactly the same, but only turn the stacking ring in the 90 degree against the Y-shaped introduction channel. So there is a change on the third dimension as Z depth, and which actually introduce the turbulence into the microfluidic channel and substantially improve the mixing process. So which actually gives a certain idea. The third dimension of the fluid control can be powerful, and it can be utilized for the certain application, especially in the biomedical research. That's some application we're going to show later. And then the other advantage of this three-dimensional geometry is really important mimicking the in-vivo cellular system. Because we know all the cell culture in the laboratory setting is through the Petri dish, the two-dimensional culture, as we demonstrate at the bottom of the figures. And the cells culture has two-dimensional Petri dish, and they show flat shape. We try to attach to the bottom and the spread and the stretch them. But in the real situation, the most serval cancer tumor cells, like we're showing here, may grow in the round shape with the cell overlay each other. So if we generate scaffolding to culture cell in the three dimensions, may grow in the morphology of the in vivo like tumor cells, as we demonstrate on the top of this confocal image. So which show the cervix tumor cells and overlay each other to form the round shape of the tumor geometry. which is very similar as the In-vivo body system. So which means the three-dimensional culture actually contributes to the In-vivo system study, in which can give more accurate study of the drug discovery or other biomedical discovery. And we know the three-dimensional is important and powerful, but how we can introduce such a three-dimensional microstructure into the microfluidics. So I'm going to introduce some methods we have developed for making the 3D microfluidic device. So here I'm showing the conventional microfluidic device fabrication, which is taking place in a clean room and need to keep the very clean environment, use very expensive facility. And one example I show at bottom the figure, which is the glass-based microfluidic chip made in the clean room. And there's some cheaper solution. Use the PDMS polymer material to make the microfluidic chip. But for the molding of the PDMS, you need to use the UV photolithography, which is a very expensive equipment set up in the clean room too. And now, along the development of the 3D printing technology, we actually can use the 3D printer to fabricate the microfluidic chip. So this is one example we on the bottom of the right hand, which is a very standard microfluidic zigzag mixing channel that prints by the 3D printer. And the 3D printer integrate all the fabrication protocols into the one step, and then you just simply convert your digital files into the 3D printing software, and then through the one button touch, and then 3D printer actually can generate the device through this layer by layer printing to form the microfluidic chip. And we use the benchtop unit, which is a customer-grade 3D printer, and we can print the mold to mold the PDMS polymer. And also, we can direct print microfluidic channel for the direct printing. We have the multiple 3D printers in our lab, and the one we use is LA-based 3D printer, which can produce the printing resolution at around 30 micron scale and the print microfluidic channel and very well. And we also have the high research grade 3D printer from the Envision Tech, and which can print the PDMS polymer materials. And we also can use the 3D printer to generate the molds, which can mold the PDMS into a small bricks with the different three-dimensional microstructures and reassemble them to form the more complicated three-dimensional channel network. So this is one example. We use a 3D printer to print microfluidic mixing chips, and also we can direct print the sample wells. And as you see from the top panel of the image, and then there's a sample well in the middle, and also this microfluidic mixer can introduce the fluid into the microfluidic channel for faster mixing through the capillary-force driving. And the printing performance and also the resolution was characterized by the SEM image shown on the bottom of the panel figures. And we print out different types of structures, include the post array and the micro well and hollow channels. And they show pretty good shape and also very smooth surface. And the printing resolution to form the hollow channel is around 100 microns. And we also test the molding process. So we use this 3D printer to print the molds, and then use the molds to mold the PDMS polymer. So as we show in the figure, we can mold all different types of the microfluidic channels and to replicate the PDMS chip from the molds and reusable for many times and then just attach this PDMS layer with a microfluidic channel to the glass slice to form this microfluidic chip. It's very simple and with low cost. We also characterize the molding precision. So we compare the channel width through the molds, and also to see how they comparable with molded PDMS. So basically, through the channel dimension around 50 micron to 500 micron, so they can replicate very well with a good reproducibility. So with such three-dimensional microfabrication skills, and then we want to utilize this skill to advance the biomedical research. The first application we employ in the biomarker detection. So as we know, there are biomarkers circulating in the bloodstream, and if we can detect certain protein, which is a biomarker indicating the disease status from the single drop of blood, that can make the diagnostics much easier and cheaper. So these biomarkers can provide less invasive, more precise diagnosis for the prognosis and the prediction. So the one application we applied in the anemia POC, the point of care diagnosis. So we know the hemoglobin, which is the protein, that's the indication of the anemia. If the level of the hemoglobin in the bloodstream is very low, then that probably will lead to the anemia. And there are some point of care devices available on the market for detecting the hemoglobin from the single droplet of blood. But the device always needs a power supply, and not possible. So it's a little relatively expensive compared to the low resource setting country or third world country. Then we introduced the 3D printing technology and to fabricate the microfluidic chips, and just with the customer-grade 3D printer to generate the microfluidic chips. So it doesn't require laboratory setting. It doesn't require the very expensive facility to manufacture these microfluidic chips. And also we can use a 3D printer to fabricate the case that can fit to the cell phone camera to host the microfluidic chips for the color scale detection. So this result can be recorded by the cell phone and analyze the data and send to the hospital. And then the hospital actually can get connected with doctor. So that reduce the visits to the hospital, especially useful for the rural area. They don't have much access or the transportation to the clinical visits. So the capillary-force drilling is very important in this development because we want to reduce the power usage. And if we can treat the surface of this 3D printing microfluidic mixer into the very hydrophilic property, and this actually can introduce the flow into the microfluidic channel automatically through the capillary-force drilling. So that means we don't need any power supply to drive the flow. And we developed the surface of the 3D printing microfluidic channel and use the saline hydrophilic coating to treat the surface. And we can see the contact angle actually change. And the last contact angle actually have much more power to introduce the flow based on the capillary force, as we show in this figure. And we started a different flow speeds at the wet length along the different contact angles. So we can treat the 3D printing microfluidic channel to superhydrophilic with the contact angle around 32. So you can see it can drive the flow very quickly into the microfluidic channel. And so this treatment actually can be simply realized by introducing the solution to go through the saline reaction. And as we see before the treatment and after treatment, the surface property actually changed through the FTIR measurements on the right panel of the figure. And we can see the 3D resin after treatment show the very hydrophilic property with the contact angle around 32, have the much more power to drive the flow into the microfluidic channel. And meanwhile, we developed reagents which can introduce the redox reaction with a single blood drop and undergo the redox reaction to produce the solution into the color display. So we characterize this color scale display and with the concentration of the hemoglobin. So as we see from this calibration curve, and along the change of the color and the concentration of the hemoglobin actually change, which is a linear response, and quantitatively give the concentration of the hemoglobin. So if we record the color of the product at the end of the mixing, and we can convert this color into the exact digital concentration of the hemoglobin and give the indication of the anemia status. Like in the red color, the hemoglobin concentration means the healthy status, and if the mixing product turn into the blue, that means very low concentration of the hemoglobin, and which actually indicates the anemia status. So we developed this point of care diagnostics kit through the simple 3D printing of the microfluidic chip, and for detecting the hemoglobin from the finger prick blood. The mixing process is very fast, within 16 seconds, and the sample and the overall consumption is very low. It's portable and transferable. And we also compare our device that measure the hemoglobin concentration with the clinical measurements using the hematology analyzer. So through the 22 human subjects, and we can see the measurement is quite comparable. And the correlation with the clinical measurement is over 19%. And we also tested the specificity and the sensitivity of this clinical test, our developed 3D printing-based POC detection of the anemia. And we can see the specificity through our device based on smartphone POC detection actually show pretty good for the severe anemia detection. The specificity can reach to the 100%. For the mild anemia detection, actually the sensitivity reached to the 100%. So overall, this technology actually introduced a low-cost POC diagnosis strategy for anemia and also for the disease self-management, which can form in one process as the standalone system. And this is a comparable diagnostic sensitivity specificity with a standard clinical hematology analyzer. The other one example I want to show through the 3D printing technology actually can introduce the new geometry for realizing the more complicated microstructure. So we can use the 3D printer to generate the molds, and the mold actually can be used to mold the PDMS. And the PDMS is a small brick molded from this mold, which we can use as the Lego. Just like you play the Lego, and then you can reassemble those Lego bricks and into the more complicated structure, which is even now be able to print it by the 3D printer. So this is the one demonstration. We have the 4 PDMS molded bricks, and to assemble from the 3-dimensional microfluidic electro-transfection system. And as we show at the bottom of this figure, it actually contains the multiple 3-dimensional channel network, provides better perfusion process for culture 3-dimensional cells. And through this molding process, you can generate a more complicated three-dimensional channel network as we illustrated in the top panel of figures. And the figure D actually is molded PDMS bricks. And you can have 4 pieces of this bricks, and the 2 assembled together from the three-dimensional cell culture system. And this electroporation cartridge was printed by a 3D printer as well, and which can use to control the electric field through the three-dimensional scanning and to perform the transfection. And the 3D cell culture actually can be performed in this device and also have a much better performance in terms of the 3D cell growth. So as we see on the right hand, on the left hand, the figure A shows the distribution of the 3D culture cell with the staining of the nucleus. And we can see the cell actually distributed in the three-dimensional culture environment in this device. And the cell grow in the three-dimensional morphology, more like the in-vivo tumor shape. And then we also characterize the 3D cultured cells by using the SEM and TEM. We can see cells grow just like in-vivo tumor cells with very good attachment and interaction with extracellular matrix. And this three-dimensional arrangement of the channel network, as we illustrated by this console simulation, provide better performance of the perfusion, and which can provide very efficiency in the supply of the nutrients and the waste exchange and also the media exchange, which allows the cell to grow much better. So as we see in the figure C, we generate the 3D microfluidic electro-transfection system with three-dimensional channel network and a diffusion from top and perfusion channel, and can go much faster and allow this perfusion happen very quickly in seconds. Much better than conventional workplace cell culture system. And meanwhile, we introduced this multi-directional electric field scanning process, in which provide very uniform electric field distribution in a three-dimensional way. And we can see that enable the plasmid to transport into the cell in a much efficiently. And as we compare at the bottom of the panel figures, if we only use a one-dimensional scanning of the electric field, and very few of the 3D cells can be transfected. But if we introduce the electric field through all dimensional, in the top, bottom, left, right, or keep rotating between the scanning, and which can generate much efficient transfection to introduce the plasmid into the 3D cells. So I'm going to play the video and in the slide 25, which shows the operation of this multi-directional electric field scanning. Okay, please play the video. All right, so you can see the scanning process because the electric field rotating between all the dimensions, so which can introduce the plasmid for transfection much more efficiently and meanwhile keep the good cell viability. So here we studied the different conditions of this electric field scanning, include the voltage, the pulse duration, and duty cycle, and also the concentration of the plasmid that we can apply. And we can see the transfection rate can reach to more than 15% at certain voltage, and also meanwhile keeps the good cell viability above 80%. So the plasmid concentration definitely is the key parameter here. And if we keep the high concentration of plasmid, and the transfection rate always can keep it high, and meanwhile keep the good cell viability. So that allows us to optimize this transfection process with good parameters to control the best efficient transfection. And we also utilize this transfection process to introduce the green fluorescence protein plasmid into the cervical cancer tumor cells, and which culture as three-dimensional culture system in our device. So we can see the plasmid was introduced into the 3D cells, and which actually grow in the green fluorescence, which can be measured by the flow cytometry. So through this measurement, we can count the cell that can be transfected, and to calculate the cell transfection efficiency and also the cell viability. And this is very useful, especially for the tissue engineering, because the cells need to be transfected with a certain plasmid to regulate tissue function. And the one application we actually employ to deliver the CRISPR-Cas9 plasmid into the HEK cells. And as we see, the cultured HEK cells in the 3D actually grow like a round shape, like in-vivo tumor shape. And we can introduce the CRISPR-Cas9 into the 3D cultured HEK cells, which can transfect it into the cells to show the green fluorescence color. And we also stain the nucleus, which is shown in the figure 1F, which is a confocal image, and can count the cell numbers within the 3D culture cell clusters, so we can see how many 3D culture cells was transfected by the CRISPR-Cas9 plasmid, and we can know the transfection efficiency. As we see from the figure D, which is the one single cells, and transfected with this CRISPR-Cas9 GFP, and when this cell divided into two daughter cells, actually they pass their green fluorescence property into their daughter cell. That means very effective CRISPR-Cas9 transfection. So those two examples shows the good application of the 3D microfluidics, and we demonstrate that 3D printing enabled microfabrication of the microfluidic device with a three-dimensional microstructure that includes the direct printing and 3D molding of the PDMS microfluidic chip, and also we can use the 3D microassembly to create a more complicated channel network. And the fabrication process is simple, fast, and low cost. And the 3D microstructures implemented into the micro device have facilitated the control of the biological system for advancing the biomedical research. All right, at the end, I want to give acknowledgement to my collaborators from the KU and the KU Cancer Center, Children's Mercy Hospital, and our states, and also want to thank the funding support and also my group member for postdoc and for graduate students here. All right, thank you very much for your time.